I think that one of the things that most people don't really know about is what the natural infection, the natural history of a viral infection actually is. And so I thought I'd go through that because they're pretty much the same for every virus. And first of all, viruses are pretty simple things. They're basically parasites, and they're not really alive in terms of how biologists describe life, because the only thing they can do is replicate themselves, but they can't metabolize foodstuffs and generate other things and so forth and so on. So they're pretty simple little machines that all they really want to do in life is make more of themselves. And they're very simple organisms in that they have a protein, lipid, fat coat, sometimes with other proteins sticking off of it like the coronavirus has. And that's why it's called the corona, because it's the crown. It looks like a crown with all these spikes coming off of it. And those spikes attach to receptors that are present on the cell surface of different kinds of cells. And each virus attaches to a different receptor molecule, some of which are expressed on lymphocytes and some of which are expressed on lung cells, such as in the coronavirus, and other cells in the body. And so you have to first, one of the things you first want to know about is what's the receptor. And in the instance of coronavirus, it's called an ACE2 molecule. And the ACE stands for angiotensin II converting enzyme 2. So just think about ACE. That's probably the easiest thing to do. Now, the thing is, the ACE molecules are distributed on lung cells, the lining of the air sacs that I talked about before, as well as the cells lining the bronchi. And in addition, it's also present on blood vessel cells, the capillary cells, and also the cells of the heart, both the vessels in the heart as well as the heart muscle cells themselves. And that becomes important in terms of the pathophysiology of the virus. It's also expressed on intestinal cells and on kidney cells. So these receptors are sort of all over the place. And so in order to gain entrance into the body, the virus has to be able to invade far enough so that it gets into the bloodstream. Once it gets into the bloodstream and it gets to the lung or to the heart or the kidney or the intestine, then it finds its receptors and then it's home. Once it's home, the membrane of the virus sort of fuses with the membrane of the cell where the receptor is present. And the whole thing becomes one. And the virus then gets inside the cell. And once it gets inside the cell, it uncoats itself, takes its coat off, and releases its genome inside the cell. Once that's there, the genes start to reproduce themselves, and each virus has structural genes that code for different proteins and so forth, so that the virus can re-encapsulate itself, reinvent itself. So once that's going on, then the virus just goes bonkers, essentially, and makes more and more and more and more and more of those of virus particles, and consequently it just sort of takes over the cell. And so the cell can't do what it normally does. But while the virus is inside the cell, parts of the virus, parts of the viral proteins and gene products and so forth, find themselves on the way to the surface of the cell. And so the viral sort of signature is placed on the surface of the cell. Now that's important because that's what the immune system recognizes. So as time goes on and the virus has been replicating in there, ultimately they get to be so many virus particles that they sort of burst out through the membrane of the cell and release all the contents of the cell. And then all those new virus particles infect other cells. So, with each particular virus, the time sequence of this whole process is different. And that is what accounts for the so-called incubation period. The coronavirus, it can be quite long, up to 10 to 14 days. And so all during that time, the virus is making more of itself, and it's just happy as a clam, and it's doing what it's supposed to do.
Fulfilling its purpose.
Right. But in the process of replicating inside those lung cells, let's say, ultimately the lung cells are damaged and destroyed. And that's a problem,
right?
And that's what I think starts the initial cough that you get, is the virus upsetting the equilibrium inside the lung. But the symptoms of the infection really don't occur until the immune response kicks in, recognizing those viral products that are on the cell surface and so forth. So then there are several different white blood cells that get activated.
Mm-hmm.
They start to congregate at the site of the viral infection, and they recognize the fact that the virus is there, and they say, we better get rid of this thing. And so they start attacking the virus and the virus infected cells, and they're killing the cells too.
Right.
So the virus is killing the cells, and now the immune system's killing the cells. It's not good for breathing. And so it's a little bit of
an understatement, yeah.
Right. But if that wasn't really bad, it gets worse, because when the immune system starts to get really revved up, it makes all kinds of molecules that bring all the powers of the immune system into the mix there. And as I said before, one of the major things that happens is that it causes the blood capillaries to leak their plasma into the air sacs inside the lung. And that's when people start to have shortness of breath. And that's because they can't keep their oxygen level up high enough, and they can't blow off enough CO2 in the whole process. And that can lead to respiratory failure. Now, before that happens, there are different things that the doctors can do to try to compensate for the fact that there's a problem down there. But the other thing that happens is during the whole process, and this is data that have come out of Italy, of the people who died in the intensive care unit, only about 50% of them really had severe lung problems, so that they weren't exchanging oxygen properly. But a lot of them had heart failure.
Okay.
And they haven't done all the studies and traced the whole thing yet. But what the thought is that the virus is binding to the ACE receptors that are also on the heart muscle and the blood vessels and so forth, and also on the intestines and in the kidneys. So that can lead to multi-organ failure, and in particular heart failure. Once heart failure sets in, then it's a problem. And the doctors have different kinds of drugs they can give to try to compensate for that and so forth. But that's really the whole picture in terms of how devastating this virus can be. And one of the things that's sort of interesting, although this virus is not a whole different from most viruses. Some people get the really full-blown, really bad disease. Maybe a lot more get pretty sick, but that they recover. Mm-hmm. Some just have a mild disease, and other ones don't have any sign of infection at all. So that's the gamut of the natural history of a viral infection. And the thing that I don't quite understand yet is that they have said that usually when the immune system starts reacting to the virus, it ultimately, if you don't die, the immune system wins.
Right.
And it gets rid of the virus. And the very almost all of the virus, and it gets rid of them, so that now hopefully your symptoms subside, and you should no longer be shedding virus anymore. And what people have said is that the virus can still be shed after the symptoms disappear in the coronavirus infection for as long as 2 to 3 weeks afterwards. Now that's a problem, because that means we have to still test people after they've recovered from the illness to see whether or not they are still shedding virus, and if so, how long, and so forth.
Right. And is that particular aspect of the virus, is that unique to this coronavirus? Does this happen with other viruses?
Not usually. As soon as the immune system, when you're having the symptoms of fever and chills and muscle aching and so forth, that's because there's a major battle going on in your body between the virus replicating and the immune system fighting it.
Right.
And if and when all those signs of inflammation go away, then the virus is usually gone. And so I don't understand what the data are. I haven't really read the primary papers, so I don't know why that happens at this stage of the game.
That is one of the more interesting aspects of it, is that from what little we understand right now, it seems to remain dormant even after you may have fought it off. Or the other thing that interests me is the whole asymptomatic part, where you have potentially a large swath of the population has had it, or has it, doesn't know that they had it, never will know that they had it unless they go and get tested for it. Is that another thing is that a common expression of other viruses as well, or is that another?
Not really. Although maybe it's because we really haven't tested. It's only in recent times that we've had these very sensitive tests for viruses and other bugs, and for the most part, they are all DNA-derived assays whereby they're very, very sensitive and very, very specific. And we've only had them, for the past 10, 20 years. And so each bug is a little different. But the asymptomatic period from the time that you initially become infected until the time that you start to have symptoms of fever and so forth, you can be shedding virus during that whole time.
Right.
And that's a major problem from the standpoint of the epidemiology and the public health end of things, because if you got people walking around town shedding virus, and of course they don't, you can't see the viral particles, they're too tiny.
Right.
But they get them on their hands, they rub their hands, their eyes, their nose, their mouth. So they've got it all over them, and anything they touch then can be contaminated.
These are the fomites that we've talked about before, The other F word.
Yeah, right. And that's why both the asymptomatic period and the fomite problem is why this thing is so contagious.
Right.
And it's really unparalleled from that standpoint. So that the initial guidelines that they told us all on the so-called social distancing, that was all based on stopping aerosols from coughing and sneezing from one person to the next person. So they've calculated that you have to, if you're 6 feet away, that the likelihood of aerosol droplets, water droplets coming, transferring to the other person wouldn't be very likely. But they didn't take into consideration the fomites, and they only started talking about it about 2-3 days ago. There was an article, I think on the 17th of March, on St. Patrick's Day in the New England Journal of Medicine, and so that was out there, and then they started talking about it. The task force. They said, "Oh, by the way, we have to worry about surfaces now."
Right.
And I thought from the very beginning, this is a major, because I read about that, and I knew that this was a problem.
Right. I remember you were ahead of the curve on that with the fomite issue. Yeah, that's very true. So I was curious to know what you thought of how the heck do I pronounce it? Chloroquine?
Chloroquine, yeah.
Chloroquine.
And
Yeah.
hydroxychloroquine. Well, I need to do more research on them, but basically these drugs have been around for a long, long time, as everyone now knows about, and used as treatment for rheumatoid arthritis, malaria, and so forth.
Right.
And there were anecdotal reports coming out of China that the patients might have benefited from these drugs, and that's what all the hoopla is based on. I think it's hydroxychloroquine that may have shown to decrease the numbers of the ACE receptors
Okay.
for the virus, and so that would give some scientific rationale to their use. But as Tony Fauci and others have said, you have to pass two tests in order to start prescribing these things for humans. One is that they're safe and safe for the particular disease you're using them for, and also that they are effective.
Right.
And in order to do that, you have to do clinical trials. The safety is done with dose-finding safety studies in people who are infected with coronavirus. So you have to start with a low dose, and then with several patients, and then if they don't die, you can go up to the next dose, and so forth and so on.
Right.
I was being facetious. You don't have any side effects. So, and then if they pass that, then you can establish a dose that is non-toxic. Then you need to treat a lot more patients with that dose to see whether or not there's any benefit whatsoever. And now we're talking about hundreds of patients.
Right.
And then if it looks like it's efficacious in those trials, then you have to do controlled trials so that you have a group of people who don't get the drug and a group of people who do get the drug, and then you give them, and then you follow them and follow their outcomes. And so all that takes time.
Right. Just the thing that we don't have.
Right.
Yeah.
So here we got this raging epidemic and so forth and so forth. So that's the story on those two particular drugs. But there's other drugs out there that are also have been tried already by the Chinese that have been given in other indications so that they're in the formulary and so forth. And for sure they should be tried, but they need to be tried in a way so that we just don't waste time, and we, at the end of their trying period, that we gain information.
So another thing that I wanted to ask you about is the discussion around the fact that you can beat this virus, but then you still potentially have the ability to get reinfected at a later date, and that you don't quite kick it in the same way that you do something else where you've beaten something, you gain some kind of an immunity. With this, it seems that doesn't really happen in that same way. So what exactly is the science behind that?
Why do you say that? I'm not sure that that's the case, really. I don't think we know now.
Okay.
Whether if you recover from an infection, whether you can be reinfected or not.
Okay.
And if that's the case, it would probably depend on the fact that the virus, that there are swarms they talk about swarms of these things. So there are multiple genes being replicated in these viral particles. And it's just like a swarm of locusts. Now, if there are some that have mutations that are particularly advantageous, they could grow better and survive better than some of their brothers and sisters. They might be more infective, and so that could happen. But I don't think it's been shown yet in this for this particular virus.
Okay, let's see.
So I'm assuming that once you do recover from one of these infections, that you will have evidence of an immune reaction. And in that instance, the kinds of things that we usually measure are twofold. One is we measure for antibodies, which are molecules that the immune system makes that bind to the particular what we call antigens, the molecules on the surface of the microbe, or in molecules inside the microbes. And so they appear in the serum of people that have been exposed and have gained have had an immune reaction to the microbe. And the other thing that happens is that the T lymphocytes, which are named after the thymus, which is a big collection of lymphocytes in the upper chest. And the thymus is important for another kind of lymphocyte. So we have B cells that make antibodies and T cells that kill other cells, and also make all kinds of other molecules that are called cytokines or lymphokines that then float around and bind to other cells, other lymphocytes, that tell the lymphocytes to become better killers or better antibody makers, and so forth. So it's a whole network of things that start to happen once the T cells and the B cells are activated. So if you recover from the infection, then you ought to have circulating antibodies, and you also ought to have a larger proportion of T cells that actually recognize this particular virus. And we can test for all of those kinds of things.
I see.
And so that brings up one of the things that I think, the president once, for example, announced yesterday that he thinks that we ought to get back to work. Mm. And of course, he's worried about the economy. And there's a problem with that, and the major problem is that we haven't been able to test all of the people in the country to find out whether or not they've been exposed to the virus, whether they are infected with the virus, whether they've gotten better and over the virus so that they have antibodies and T cells reactive to the virus.
Right.
So we don't know who's naive, so to speak, so that has never been exposed to the virus, or who's infected, or who's immune.
Right.
And if we then tell everybody to go back to work, we're just going to be stirring the pot.
Right.
And it's just going to get worse. And so that's a major problem, because if it makes it worse, then the president's going to be very unhappy because the economy is going to get worse and worse and worse.
You just created a bigger problem. Right. Yeah, we talked about... Oh, go ahead.
Yeah, so what we need what they call point of care tests for the virus on the one hand, and tests for antibodies to the virus on the other hand, because we need to be able to discriminate between those who are infected versus those who are immune. Now, we also need to go not to the whole population of 360 million Americans, but to samples of different, parts of different areas of the country, like New York and Seattle and Chicago, and also areas of the country that have not had a lot of cases yet, to find out what the prevalence is. We need to test all the people, let's say 1,000 or 2, just a random sample. And
per each of those areas, you're saying? Yeah,
for example. And we need to do all the areas in the country, but those are a place to start, to find out what the prevalence of the virus in the population is. Is it 1%, or is it 10% of the people that have it, or is it 20% of the people that have been infected? Because those numbers will then tell us what our job is, essentially,
right?
And how easy it would be to restart the economy in that kind of an instance. And this is what China did in I think the Hebei province, and what Korea did. And in particular, they are the model system, because they were testing people at the rate of 10,000 a day when we only had a few tests positive, essentially.
Right.
And so they tested all the people, and people that were positive, they quarantined those people. And of course, China did exactly the same too. They had quarantine. Even if you were asymptomatic, you got quarantined to the dirty population, and you got
Right.
placed in a conveyance that you were held there until you got better or worse, and then you went to the hospital if you got worse. We can't open up the economy again, the society again, if we don't know who's infected versus who's not infected, and that's the problem. And we'll see what happens. But what I fear is that because of the political and business and economic considerations, is that they're going to make the same mistakes that they did in 1918 when they just perpetrated the epidemic. And the epidemic went through a couple of years.
There were several waves. You're talking about the Spanish flu. Yeah,
exactly. Yeah. But incidentally, there's a great book. If I didn't mention it before,
I think you did. But what is the title again for anyone interested in reading?
Well, it was just they interviewed the author on NPR this morning on the morning show. On point, and the author of that book, it's called The Great Influenza. And the author's name is John Barry. He's an epidemiologist at Tulane Medical School down in New Orleans. And I'm actually rereading that book now. Okay. And I read it about 10 or 15 years ago when I was heavily involved in flu research and so forth. And it's just a fabulous book. And I think everybody should be in the whole United States of America should read this book now.
Okay.
Because it tells you what's going to happen.
Okay. I will. I'll put a link to it in the description for this one.
Okay.
So The Great Influenza, it's called.
Yeah.
Okay. By
John Barry.
Okay.
Barry.
What else has come to your attention? What else do you feel the need to speak about in regards to coronavirus and this whole situation?
Well, there's several things actually that relate to immunology and cytokines and that sort of thing. One is the people that have gotten very sick in the intensive care units are usually on life support and on respirators, so that they have an endotracheal tube down into their lungs, and they are if they're not comatose, they're made comatose through medicines. And so these people are really on death's door. And what happens to these people, in part because of the fact that they're not exchanging oxygen very well, that their other organ systems like the kidney and the heart and the brain and the liver start to fail. And one of the things that they have started to use in tumor immunotherapy now is are medicines that will block the inflammatory response in a big way. And doctors now have started calling these situations whereby there's a huge inflammatory response. They've traced that to a huge increase in cytokines, and so they've called this a cytokine storms.
Right.
And there's one drug in particular that's new on the horizon. It's only been in use for about 10 years, that blocks interleukin 6, which is one of the major purveyors of inflammation. There's two other cytokines that are involved. One is called tumor necrosis factor, and we have drugs to block it. And another one is called interleukin 1, and we have drugs to block it. And I think all of those drugs that block IL-1, IL-6, or tumor necrosis factor will be used and will be very beneficial for the really sick people in the ICU. So
I know that we've talked about this, and a cytokine storm is what exactly? What does that mean in the human body? Is that the immune system overreacting to a perceived threat?
Well, actually. Yeah, what happens when there's a site of inflammation? Let's say you cut yourself on your arm and the bleeding stops, and you have a wound there. Over the course of the next several days, you can look at your wound, and it changes in its character. And what happens is that it gets a little bit swollen, it gets red, if you feel it, it's warm,
right,
and it's tender. So those four things are the cardinal signs of inflammation: red, hot, swollen, and sore. All of those cardinal signs of inflammation are caused by these cytokines. And what the cytokines do, the reason it gets swollen is because the capillaries start leaking their blood plasma into the tissues, because in the plasma are a bunch of other molecules that fight off the infection or fight off all of the things that are damaged in the tissues. So it gets swollen, and the capillaries and the other blood vessels become dilated, so that now that's why the color of the skin changes to red or pink. And it gets hot because there's a lot more blood flow to the area. And it's sore because some of the cytokines that are released trigger pain receptors in the nerve receptors in the area, and go up to our head, and so that's how we recognize the fact that we've got a problem there. And if we didn't have the pain associated with the inflammation, we might just sort of ignore it, and it's no problem as long as
the problem would get worse. Yeah,
right. And so now that's on a small scale, and that's all good. But if that happens in the whole body gets red, hot, swollen, and sore.
Right.
And particularly as the capillaries leak their plasma from inside the blood vessel to outside the blood vessel, the blood pressure falls. And as the blood pressure falls, you go into what we call shock. And if it isn't counteracted medically by therapy, you die pretty soon. So the cytokines are good on a local thing, but on a systemic, total body level, they're not a good thing to have happen to you.
Right. Okay.
So that's just one aspect of the immunology end of things. Another one that I've been thinking about actually, and I was working on when I was working on flu. It'd be nice to have most vaccines are designed so that the immune response is skewed towards the antibody end of things rather than the T cell end of things. If there are a balance of those two kinds of immune responses, and which is fine because the antibodies are circulating in the blood, and as soon as if there's reintroduction of the same bug, the antibodies, and that's one of the reasons that the capillaries leak the plasma, because the antibodies are in the plasma. They go to the site, they bind to the antigen, and they help other cells then get rid of that whole thing. And so there's sort of a first line of defense, the antibodies in the blood. However, particularly with viral infections that are intracellular infections, most of the bug is inside the cells.
Mm-hmm.
And so antibodies are not going to do any good for that. If the infection gets going. So T cells then are brought into the process, and T cells start killing off any infected cell it can find.
Right.
And T cells really are the reason you ultimately recover from a viral infection. If your T cells are deficient for some reason, and one of the major reasons nowadays is AIDS, because the HIV virus infects the T cells.
Mm-hmm.
And then it sets up a persistent viral infection. And so that's why people with AIDS die of infections, but they don't die of the HIV infection. They die of all kinds of other microbial infections.
It's essentially turning off the immune system's ability to respond.
So that's, and all of these bugs have evolved together with not us, other animals out there that have immune systems. It's been a co-selective process throughout evolution. And so I always say, well, thank God for Darwin, otherwise we wouldn't understand anything. That reminds me when I would get new people into the laboratory, new students, I would tell them that I'd say, we don't stop working during the holidays. We work straight through. We don't take any time off for Christmas or New Year's. But what we do is we celebrate Darwin's birthday, which is the 12th of February. He was born on the same day, 12th of February in 1809, as Abraham Lincoln.
Wow. And
so both of those guys were not bad,
So it's a good day to celebrate.
So what it'd be nice to do would be to try to make a vaccine that didn't depend on an antibody response, but would activate T cells to generate, so that you would generate lots and lots of T cells to a particular bug, like say coronavirus. And what you could do would be you could select out parts of the coronavirus, so COVID-19, that were conserved in other coronaviruses. And so it would be, what you might be able to do is to make a coronavirus pan-specific vaccine that would activate T cells so that you would be immune because of that. And then you might get rid of an awful lot of the common cold, which evidently coronaviruses cause.
Huh. That would be interesting. Is there any research going on currently that is happening around that topic now that you're aware of?
Not that I'm aware of, actually.
Okay.
So yeah, I've been tweeting about that. So I've been trying to activate the next generation.
I hope that they take the torch. What is the limiting factor on testing? Like, why isn't it as widely available as it should be? Is there something to that, or?
Yeah, well, I don't know what the problem has been, and
okay.
It's not out there in the media yet. But there, as far as I can see, there was just a major screw up, and a major screw up at the CDC in terms of decisions made or not made when they should have been made to really ramp up. And usually the CDC is the first government agency that begins to work on this kind of a situation. And in particular, because we've already been through both SARS and MERS, which were similar respiratory illnesses, severe too. The mortality rate with SARS was, I think, close to 40%, or maybe that was MERS. Yeah, I mean, they were able to contain the epidemic, but of the people who got it initially and so forth, a high proportion of them died. And at the time, and that was like 2004, and then I can't remember, 2014 for the second.
2010, I believe. Or that was
okay.
H1, that was H1N1, I think.
Yeah, and so it's not that they didn't know about these things. They were out there, and not only that, a lot was known about coronaviruses in a very general sense before those two things happened. So somebody really dropped the ball at the CDC, and so evidently the Chinese told the world about COVID-19 on the 3rd of January. So right after that, in the whole month of January, is when the CDC should have worked up a test, and there's not only work up the tests and make sure that it works properly, but then you have to then produce it in large amounts of the test and distribute the tests to the various, and they have about 100 labs all over the country they got a test, but then they had to recall it in February. and because they were getting false positives. And the intricacies of that haven't been made public, and probably it's going to be one of these things so that an enterprising young journalist will ferret around and find out all the little details of who did what, when, and why not, and so forth.
Right, and that'll be another book 10 years from now.
Right, if we all live through this whole thing. And so now with according to to Tony Fauci and the president and the rest of them, they've gotten the commercial labs. They can ramp up production very fast, a great deal. And so eventually they're going to be able to test, and they should already be testing in a sense to be able to get sample the population to find out what the prevalence of the virus is. Mm-hmm. And they can really, and they're probably doing that now. So what is it? It's towards the end of March, and so now we're sort of
it's the 24th,
3 months out.
Yeah. So
if we don't do that, we can't do anything. But public health wise, economy wise, societal wise. So we've got to have those tests.
Yeah, I mean that's part of the conversation now is how do you put a dollar amount on someone's life? Essentially, you have that double-edged sword of you either say go back to work or focus on health to the exclusion of the economy. It's a Hobson's choice. There's no choice to be made, really.
Yeah. Well, see, I'm a grandfather, so I don't want to be cast aside, and as the roaring train goes through the station.
Exactly. So here's a question for you as someone who's developed experiments their entire life. How would you structure this testing to be as efficient as possible? So you mentioned, gathering like a sample size of 1,000 individuals from each state, say. Are you looking at people that have shown symptoms already, or are you just sort of randomly selecting from the population? I mean, how do you
Yeah, no, that's what you really want to know is, as I've said, is the prevalence. And so you just take everybody. You take a random sample of the population and to find out how many have infection, and then you ask them at the same time whether they've got any symptoms. So you know that the proportion that are asymptomatic and also the proportion that are symptomatic.